Hello students and welcome to the study portal. Hope you are learning with new dimensions. To help you in that, we are here with one more video on the topic blood group system. So let's start the video. So in today's session, we are going to study about the human blood group system. So in today's session, we will be learning what is the type of blood and how it is identified. What are the reasons why the people are unique and different in themselves? So let's start today's session. So first of all, let me tell you what blood group system is. So the term human blood group system is defined by International Society of Blood Transfusion. It is defined by the ISBT, which is termed as International Society of Blood Transfusion, as systems in the human species where cell surface antigens, in particular those on the red blood cells, are controlled at a single gene locus or by two or more very closely linked homologous genes with little or no observable recombinations between them. So we can say that the blood group system is a set of antigens and antibodies that are present on the red blood cells which are controlled at a single gene locus or by two or by more very closely linked genes present on that chromosome number. Alright, so here you get the definition of blood group system, that it is a system of, or it is a subset of genes, antigens and antibodies that are present on the surface of the RBCs. Alright, so the blood group system was discovered by Austrian scientist Sir Karl Landsteiner in 1900. He discovered that human blood possesses different antigenic and immune properties. Alright, so the focus of his observation was the clumping of the blood, as it was an immunological reaction. In 1930, Sir Karl Landsteiner got the Nobel Prize in Physiology and Medicine for the discovery of blood group system. Hope this is clear to you. At present, if I talk about, there are around 38 blood group systems that have been discovered, and around 400 plus antigens are said to be present on the human RBCs. So we can say that blood groups are antigenic determinants on the surface of blood cells, but the use of the term is generally restricted to antigens on the red blood cells. So we can lead to a conclusion that a blood group system is one or more blood group antigens encoded either by a single gene or by a cluster of two or more closely linked homologous genes. Alright, so some systems contain only one determinant, while others blood group system may have more of the determinants. Alright, so we'll be studying about these blood group system. So here you can see the classification of blood group system as per the ISBT, the International Society of Blood Transfusion. So the blood group system can be studied as major blood group system, which includes the ABO blood group system and RH blood group system, which is called as the rhesus blood group system, and there are minor blood group systems, which are Kell, Duffy, Lutheran system, Lewis, Digo, Kidd, Bombay blood group, and many more. We are going to study about all the systems. So in today's session, we'll be talking about the ABO blood group system in detail. Now talking about the principle of blood grouping, that how blood grouping is performed and what is the basis of performing the blood group system. So The blood group of a person can be identified or can be performed on the basis of agglutination reaction, on the basis of the observation of the agglutination reaction. So we can see what does agglutination means now. So agglutination means the collection of separate particles like RBCs into clumps or masses. What happens when we observe the blood group system on a slide or a tube, glass test tube or plastic test tube? We mix the blood that has to be tested with anti-sera. So what happened on the basis of this immunological reaction that is going to happen because of presence of antigen and antibodies, antigen on the surface of RBCs and antibodies in the anti-sera, an immunological reaction occurs and it results in the clumping of the RBC, which can be seen as these granules or the clumping or in the masses. So based on this clumping reaction means the agglutination. So when does agglutination occurs? So agglutination occurs when an antigen is mixed with its corresponding antibodies. The corresponding antibodies are also called as the isoagglutinin. This term was given by Sir Karl Landsteiner. What does it means? That when an antigen is mixed with anti-A or when B antigen is mixed with anti-B, it will give an agglutination reaction based on which we can determine the blood group of the person. So this is the basis of agglutination reaction. Now, what made the basis of agglutination reaction that it will determine the blood group system? So when Sir Landsteiner discovered the blood group system, he gave a law which is called as the Landsteiner's law. So according to Landsteiner, if an agglutinogen is present on the red blood cell membrane, agglutinogen here means the antigen. If an antigen is present on the red blood cell membrane, the corresponding agglutinin must be absent in the plasma. Understand it with the help of an example. Let's say a person has a blood group A. So he must be possessing A antigen on its surface or on the surface of its red cell membrane. So according to Landsteiner, he must not be having anti-A antibodies, which are the corresponding antibodies. They are not present. So, and the second law is, if an agglutinogen is absent on the red blood cell membrane, let's say that the blood group of a person is A, so he must not be having B antigen on its surface. Alright. So if he is not having B antigen, he must be having the corresponding antibodies to this B antigen, which is anti-B. So this makes the law or the basis of agglutination reaction. We'll be understanding it deeply in the later sections. So this is the Landsteiner law. Hope this is clear to you, that if an antigen is present on the red blood cell membrane, the corresponding agglutinin or isoagglutinin must be absent in the plasma. He is not having the corresponding antibodies to that antigen which is present on his red blood cell membrane. Let me make it very clear to you that the blood group of a person is determined by the presence of antigen or antibodies on the red blood cell membrane. For example, this is a red blood cell of a person. So if he is having A antigen attached to its red cell surface, then the person is said to be having A blood group. If the B antigen is present, then the person is said to be having B blood group. If both A and B antigens are present, then the person is said to be having AB blood group. And if both of the antigens are absent, then the person is said to be having O blood group. This is the very basic law, but we will understand the ifs and buts in the later section of this video only. So stay tuned. Alright students, let's move. Now the ABO blood group system. We will talk about the discovery of ABO blood group system, the antigen and antibodies involved in the ABO blood group system, and its inheritance, that how it has been inherited genetically into the humans. Moving on. Now what led to the discovery of ABO blood group system? Let's understand. So the ABO blood types were first discovered by an Austrian physician, Karl Landsteiner, in 1900. He was working at the Pathological Anatomical Institute of the University of Vienna, which is now called as the Medical University of Vienna. In 1900, he found that red blood cells, the RBCs, would clump together when mixed in test tubes with sera from different persons, and that some human blood also agglutinated with animal blood. So he gave a conclusion that the serum of healthy human beings not only agglutinates animal red cells, but it also often those of the human origin from other individuals. He concluded that it remains to be seen whether this appearance is related to the inborn differences between individuals or it is the result of some damage of the bacterial kind. So this was the first evidence that blood variations exist in the humans. It was believed that all humans have similar blood earlier. But after the discovery by the Karl Landsteiner that the clumping can be seen or observed when the RBCs of the humans are mixed with the sera of different blood of the persons or the animal blood. So there was the presence of some antigen that can lead to the clumping or immunological reaction. So the next year in 1901, he made a definitive observation that blood serum of an individual would agglutinate with only those of certain individual. Based on this, he classified human blood into three groups, namely group A, group B and group C. He defined that group A blood agglutinates with group B. Similarly, group B blood agglutinates with group A. Group C blood is different in that it agglutinates with both A and B. So this discovery of blood groups, for which the Landsteiner was awarded Nobel Prize in 1930. Alright, so then in 1910 in 1902 von de Castello and Sturley discovered the AB blood group system also to this blood group system. Thereafter, in 1910, Ludwig Hirschfeld and von Dongern introduced the term O null group for what? For the group that was designated as C by the Karl Landsteiner. They were also the first to explain the genetic inheritance of the blood group system. So the discovery is clear to you that in the earlier stages, who gave the classification? First of all, Sir Karl Landsteiner discovered that there is some clumping that can be seen with the RBCs of the human when it is mixed with the serum of different persons and the animals also. Thereafter, he led to the discovery that the agglutination is happening when with the certain serum only, not with every blood group, not with every blood or every blood type. Then in due to this observation, he gave three kind of blood group system: A, B and C. That the A is agglutinated by B when mixed with B, then B is agglutinated when mixed with A, and there is a third blood group system also, C, which agglutinates with both A and B. Thereafter, in 1902, von de Castillo and Sturley discovered that there is one more system which is called as the AB blood group system, which contains both A and B antigen. Thereafter, in 1911 or 10, the von Dungen and Ludwig Hirschfeld divided the group A into two subgroups, A1 and A2, and also given the name O null group to the C blood group system discovered by the Karl and Steiner. Moving on. Now, and talking about the antigens of the ABO blood group system. Alright. So, the antigens of the ABO blood group system appear in the 6th week of the fetal life. They are present on the red blood cell membrane and in other tissues also, like salivary glands, pancreas, kidney, and except the CNS. It is not present in the central nervous system. So there is H antigen, which is also present usually in all individuals. We can say that H antigen is the precursor of A and B antigen. Moving on. Now talking about the biochemistry of this A, B and H antigens. So the A, B and H antigens are present on the red cell surface membrane on the locus of some genes on different chromosome number. Alright. So these are formed by either by type 1 chain beta 1, 2, 3 linkages or by type 2 chain beta 1, 4 linkages. So the precursor substance stays the same. The precursor substance of these antigen is glucose, galactose, N-acetyl glucosamine and galactose. The ABH antigens are formed by these precursor which are there for ABH antigen. So this precursor substance remains the same, plus other group get attached and it leads to the formation of either A, B or H antigen. Alright, so let's understand. So now talking about the formation of H antigen, that how H antigen is formed. The ABO gene resides on chromosome number 9 at the band 9q34.2 and it consists of 7 exons. It is remarkably to understand that the ABO gene is located on the long arm of chromosome number 9. The ABO locus encodes 3 alleles, that is 3 variants of the same gene. One allele is derived from each parent. The 3 alleles are A allele, B allele and the O allele. So the A allele produces alpha-1,3-N-acetyl galactosamine transferase, which catalyzes the transfer of GalNAc residues from the UDP GalNAc donor nucleotide to the Gal residues of the acceptor H antigen, converting the H antigen into A antigen in A and B individual. Then the B allele, it encodes alpha-1,3 galactosyl transferase, which is also called as the B transferase. The difference between A and B glycosyl transferase enzyme is of only 4 amino acids. Whereas if we talk about the O allele, it lacks both enzymatic activities because of the frame shift which is caused by the deletion of guanine 258 in the gene, which corresponds to a region near the N-terminus of the protein. Now, let's understand the formation of H, A and B antigen to understand this concept of alleles better. So, first of all, you have to remember that the genes of the A, B and H A antigen are located on the long arm of chromosome number 9. Alright. Now talking about the formation of H antigen. As I told you in the previous slide that the precursor remains the same for A, B and H. What is the precursor? That due to the red cell membrane, glucose, galactose and N-acetyl this glucosamine is common. Alright, you can see the further picture also, that glucose, galactose, N-acetyl glucosamine and galactose is common. This is the precursor substance at it, and it remains the same each time. So, which leads to the formation of H antigen. The addition of group fucose leads to the formation of H antigen, and then it is termed as L-fucosyl transferase enzyme. This is a product of H gene. So, due to the presence of this enzyme, a fucose group gets attached with the precursor substance in the red cell surface membrane, which leads to the formation of H antigen. Moving on. Now, what leads to the formation of H antigen? To this group, N-acetyl galactosamine is attached. So, this is common, and N-acetyl galactosamine gets attached, and it leads to the formation of A antigen. Alright, so A antigen is termed as N-acetyl galactosamine transferase, which is the product of A gene. Now, the formation of B antigen. To the whole group of the precursor substance, galactose get added, and it is termed as D-galactosyl transferase, which is a product of B gene. With the help of this enzyme, galactose gets attached, and it converts the H antigen into the B antigen. So, these are the difference between A, B and H antigen. Now, talking about the O gene. You have heard that the O gene does not consist of any antigen such as A or B antigen. The O group does not have A and B antigen, and I told you that there are three type of alleles that are present on the surface of that may be present on the surface of red cell membrane. These alleles are A, B or O allele. So, on the basis of that, a person's antigens are determined. So, why do group O individuals have more H antigens than the other group? Well, let me tell you one more thing, that if a person is having A antigen or A blood group, then he must be having A and H antigen. If the person is having B blood group, then he must be having B and H antigen. And if he is having O blood group, then he is having H antigen, not A and B. So, why do group O individuals have more H antigens than the other group? Because the O gene is a silent allele. It does not alter the structure of H antigen, H substance. Because it is silent, it is not dominant, thus it does not alter the structure of H antigen. Whereas the A and B alleles are dominant. So, it alters the structure of H substance, and it has fewer, and they have fewer H antigen sites, and A antigen gets attached with it in the A group, and B antigen get attached in the B group. But O allele being silent, it does not alter the structure of H substance, due to which there are more H antigen sites. Thus, the H group H antigen attaches to the more of the sites, leaving only some space for the other antigen to attach. Therefore, the H antigen does not have the presence of A or B antigen in it. Hope this is clear to you now. Now, talking about the antibodies of the ABO blood group system. So, mostly the antibodies of ABO blood group system are of IgM type. It consists of immunoglobulin M type. These are produced by the bone marrow and the lymph gland cells, and they are also known as the cold antibodies. So, these are the average sites of A and B agglutinins in the plasma of people with different blood types. So, it reduces as the age of the person increases. The average titer, it decreases as the age of the person increases. Moving on. Now, why antibodies produced in people who do not have the respective antigens in their blood cells? As you can see that the person having O blood group does not have any antigen, but they have antibodies, anti-A and anti-B, both antibodies. So, why these antibodies are produced in people who do not have respective antigens in their RBCs? There arises the question. So, the immune system form antibodies against antigens that are recognized as not self or non-self, that are recognized as the foreign for the body. So, they are not present in the own body, but sometimes due to the genetic exposure, we get exposed to these antigen. Thus, our body produces certain kinds of antibodies. Alright. Thus, we can see that if the certain antigen is not present, then also body has antibodies. Now, you can see the types of ABO blood group system. So, here you can see there are 4 type of blood group in the ABO blood group system. First one is the A blood group, B blood group, AB blood group and the O blood group. And there is the prevalence of the blood group system which can be generally observed. So, group B is mostly seen in the people. It is mostly present, and group AB is rare. Alright. So, what made the differentiation between these blood groups? We can say that the presence of antigen and antibody differentiates the blood type of the person. If the person is having A antigen on the red cell surface, then the person's blood group is said to have A blood group system, and he must be having the different antibodies such as anti-B antibodies, because according to the Karl-Landsteiner law, as we have studied, that if certain antigen is present, then he must not be having the corresponding antibodies. So, he is having the different antibody. Alright. So, group A, the blood group of the person is determined by the presence of antigens on its red cell surface. If the person is having B antigen, then the blood group of the person is B. If the person is having A and B both antigen, then the blood group is AB. If the person is not having A and B antigens, he is just having the H antigens on its red cell surface, then the person's blood group is said to be O, which is also called as the null blood group, because it does not have any antigen. Alright. So, whatever antigen you are having, your serum will having the different antibodies for it. Suppose if the person is having A antigen, then he must be having anti-B antibodies. If the person is having B antigen, then the person is said to have anti-A antibodies. If both A and B antigen is present, then none of the antibodies will be present. And if the person is having O, which is none antigen, then the antibodies will be anti-A and anti-B. Hope this is clear to you now. Now, talking about the inheritance of ABO blood group, that how we inherit the blood group in the in our blood. Alright. So, the inheritance of blood group, ABO blood group, is followed by the Mendelian law. In 1924, the Felix Bernstein proposed presently accepted theory of the inheritance of the blood group system, that the blood group system is determined by the codominance, dominance or the recessive type of alleles present in the parents. So, the blood group of the person is determined by its parents. It is given by the parents only. Whatever blood type the mother and father have, based on the alleles present as dominant, codominant or recessive, the child or the offspring get their blood group. So, if the father is having blood type A and the mother is having blood type B, then the father must be having A and O alleles. whereas the mother would be having B and O alleles. So based on the dominance, codominance and recessiveness, their offspring may be having A blood group, B blood group, AB blood group and O blood group. This picture represents the codominance, recessiveness or the dominance of the person. That if the child receives a dominant A allele and recessive O allele from the mother, then the child will be having A blood group. If the child will receive both dominant alleles, A from the father and B from the mother, then the blood type will be AB. If the offspring receives dominant B allele from the mother and the recessive O allele from the father, then the blood group will be B. If the child will receive both the recessive allele, that is O and O, null, then the blood group system will be O. So based on the dominance, codominance and recessiveness, inheritance of the alleles, the blood group or the blood type of the person is determined or inherited. These are the examples. You can see that if the mother is having AA and father is having BB allele, then the offspring will be 100% having the chances of getting AB blood group. If the mother is having BO and the dad is having OO, then there is 50% chance of each B blood group and O blood group. If both of the parents are having O blood group, then there are 100% chances of offspring having O blood group. If the mother is having O blood group and the father is having A blood group, then there are 50% chances of each having A and O blood group. So that's all about the ABO blood group system. Hope this has helped you in understanding the ABO blood group system in a better and more systematic way. Please stay connected and watch your videos, your LMS regularly to attain good knowledge of all your subjects. Thank you, students.